Go ahead, Jihad.
Hello, everyone. Can you hear me? Am I audible?
Yes, go ahead.
Okay. Yeah. Welcome, everyone, to this workshop. Today, we'll be discussing the theory behind molecular docking and hopefully the practical, the practical part of molecular docking. We apologize. We had some technical issues with the laptop that has the docking files, and until now, we're not very certain whether we'll give the practical session today or tomorrow. If the technical issue persists after the theory session, we'll decide upon it. Anyways, we can start now with the theoretical introduction. We have some learning objectives. Introduction to theoretical principles behind molecular docking and understanding ligand-receptor interactions, to be specific. We'll have a quick tour over the databases, the auxiliary tools, the helping tools in molecular docking. And we will have an overview over the different docking software, including AutoDock, which we will work with on the practical session. Practical docking workflow, hopefully today or maybe tomorrow, and some further insights and applications. What is behind molecular docking and what's next? Hopefully in the upcoming workshops, we'll have an introduction to, for example, molecular dynamic simulations, allosteric binding docking, and so on. Okay. So we usually in the experiments we do in life science experiments, biological experiments, we usually look at high-level observations, for example, looking at the oxidase test in microbiology. We have the catalase test, positive, negative. We have changes in colors. We have certain assays. But what is actually underlying the such processes at the molecular level? So actually, that's one of the the goals of molecular docking is to understand basically the molecular interactions governing the cellular processes, and maybe molecular interactions. It's not just limited to pharmacology. It's not just limited to biomedical research. Molecular interactions are everywhere. Maybe in environmental sciences, we want to design certain enzymes to digest certain molecules. But today we'll have more focus on the pharmacological aspect of molecular docking. So for example, this is a crystal structure of a sugar with the glucose oxidase responsible for this reaction. So we can understand how the glucose oxidase binds with the substrate and what's the posing orientation and the mechanism, and some insights to the mechanism. In chemistry, you might have had an introduction to the different types of molecular interactions, including intermolecular forces and intramolecular forces. So we have forces within a molecule and we have forces between the molecules. For example, in proteins, when the protein folds from the primary structure to the secondary structure, tertiary structure, different intermolecular interactions take place, including hydrogen bonding. We have a polarized OH bond, and we have a kind of hydrogen bonding. We could have ionic bonding between a cation, anion, and a cation. We could have disulfide bridges and disulfide bonds, which are covalent in nature, and sometimes dispersive forces, dipole interactions, maybe temporary dipoles, maybe permanent dipoles, and so on, which are the induced forces. And we can see that different forces have different strengths. Usually the intramolecular forces are strong, relatively strong compared to the weak intermolecular forces. For example, when talking about covalent bond, strong ionic bonds, in comparison, for example, to the van der Waals interactions or the dispersive forces or the dipole interactions, they are weaker. And these are very important in molecular docking. We want to know what kind of force fields we have between the ligand and the receptor. Anyways, we have here, for example, an example of a protein ligand binding to each other. We have controls, we have the molecule, and we can see the landscape of the interaction. It's not a chemistry class, but this was a gentle introduction. As we can see, we have the intermolecular forces between water molecules and the intramolecular covalent bonds within the water molecules. We have also other properties in chemistry. We have the stretching of bonds, the length of the bond varies. We have bending of the angles, torsional forces and torsional rotations can take place at certain bond, and even non-bonded interactions, non-bonded intermolecular interactions. So having some biological bases, we know that we usually have a binding interaction between a molecule. Usually it's a small molecule ligand and an enzyme protein, which can result in activation, inhibition, agonism, antagonism, and so on. And this underlies lots of reactions in our bodies and lots of responsive mechanisms in our bodies, whether it be a receptor-ligand interaction, whether it be a substrate-enzyme interaction, and so on. And we'll see what kind of interactions molecular docking can give insight to later. So looking at the models of action, usually in enzymes, we have different models that can explain such interactions. We have the rigid lock and key mechanism. For example, the substrate is the key, for example, and for example, the enzyme is the lock, and you know they have a highly complementary structure, and they bind to each other in a rigid fashion. Sometimes it could be more flexible. We could have induced fit mechanisms where there is a change in the shape during binding and after binding. And we have also the transition state model, or the transition state mechanism, in which we have a high energy transition complex state. And after that, we will have the product formation from the substrate. And within the enzyme, we could have different binding sites. We have the active sites. We have sometimes allosteric modulation and allosteric site modulation, such as in hemoglobin protein. Sometimes we could have single and multi-chain binding sites, or sometimes cryptic binding sites. These cryptic binding sites, sometimes they could be hidden within the molecule, and sometimes they could be exposed upon binding of the ligand and not in the naive state of the enzyme. So we can see here the protein structure. We have the active site, binding site. We have the catalytic site. The binding site is for the binding orientation of the substrate. And here we reduce the chemical activation energy, which is the main mechanism of enzyme-based catalysis, which reducing the activation energy for the reaction. The ligands can be different biomolecules. They could be exogenous, endogenous molecules. They could be synthetic, they could be natural. So they could be proteins, they could be nucleic acids, they could be metal ions, cofactors, water, glycans, and so on. The molecular mechanisms of drug actions. So as we see, most drugs are endogenous compounds. A drug is simply something that we administer and it produces like an effect, a biological effect. It can change, it can bind to certain molecules, it can compete, it can inhibit, it can block, and so on. So, for example, we could have drugs that modulate ion channels. We could have drugs that agonize, antagonize receptor, and thus affecting further signal transduction pathways. We could have enzyme inhibitors. We could have pro-drug on enzymes, and so on. That's not our main focus today. The potency of drugs can be assessed by certain kinetic parameters, including the inhibitor constant. Inhibitor constant, which is an indication of how potent the inhibitor is. It is basically the concentration that is required to produce half of the inhibition. So a lower Ki or a lower inhibitor constant means a more potent molecule. And the Ki can be calculated also from some thermodynamic quantities, including the Gibbs free energy, and we have these RT constants. So we could imagine a more negative binding energy change, a more negative Gibbs free energy change. It will indicate, for example, a lower Ki and thus a higher potency and higher binding affinity. So as we can see here, larger inhibition constant means lower binding affinity, and vice versa. And this is important because in as we will see in the practical session, that from certain estimations, we can have like a theoretical estimation of the Ki of such interactions. We can see here the binding affinities. Here we have, for example, the percent receptor occupancy, which could be directly or indirectly translated to, for example, in terms of enzyme inhibition, for example. So we can see here at 50% occupancy or maybe 50% inhibition. We can see the drug concentration, the corresponding drug concentration, which is equal to the constant, the equilibrium constant, which can be Ki, as we said. In molecular docking, usually we follow like a reverse pharmacology approach, which is like a target-based approach. We identify proteins. We usually have a couple of candidate compounds, like we screen for several compounds, whether they interact or not with the target protein, and how favorable is such interaction. And we then assess the functional activity within like in vivo models. And in forward pharmacology, we simply just assess the activity of, for example, such enzymes or such proteins, and for example, diseased models. And we can identify it as a target. So we basically, in molecular docking, we follow this reverse pharmacology approach. We screen chemical libraries, like large libraries of small molecules or candidate drugs are screened to identify compounds that bind favorably and with high affinity to the target. We usually have like this kind of hit identification. We could have like hundreds of compounds, and then we can shortlist them to 10 and compare them. So this is it. So we begin with the target. We identifying and targeting a specific protein that is believed to be disease modifying. So it's hypothesized, or it's hypothesis driven process, as we can see here. So what is molecular docking? Simply is a very important tool in structural biology. In general, or the two main questions that we answer in molecular docking: do they interact, and how favorable is this interaction? As I said, which the favorability of such, for example, interaction can be assessed by, for example, the energy change, the binding affinity, and so on. What orientation, what relative orientations of the docked molecule and the ligand correspond to this lowest energy state, or this most favorable interaction. So what orientation maximizes interactions or minimizes the energy? What orientation is the most favorable? So from molecular docking, we can get insight on the specific exact landscape of bonding taking place between for example, the receptor and the ligand, and we can do, for example, pharmacophore modeling, and this can give insight into the function associated with such structure. So it is structural, but sometimes with further modeling, further pharmacophore modeling, we can identify certain biological mechanisms underlying the action. So we have some terminologies. We have the receptor molecule, we have the ligand, or for example, the key. In the case, for example, of the lock and key model, we have the process of docking for sure, the binding mode or the binding mode, and we have the pose, which are similar. The pose, which is the specific orientation we're looking at. We have the scoring, which can correspond to the, for example, the energy change, and we have the process of ranking the molecules based on a certain scoring function. So basically, molecular docking looks like we have to prepare our ligand, we have to prepare our receptor. After that, we define our docking problem. Is it rigid docking? Is it flexible docking? And we'll see later. And then we'll generate the poses. We'll score each pose and we'll rank them. And then we can compare them. We can do further analysis, such as dynamic. Like, for example, when we deal with molecular docking, it is a static process. Sometimes we could extend it further. So it's a molecular dynamic simulation, which could be more insightful in terms of theoretical measurements. And sometimes we have to validate our findings. In the case of a novel interaction, we have to validate our finding, whether it be using crystal structure or further, for example, experiments that can knock out certain interactions, and so on. So as you can see here, we have the molecular dynamics. We have the binding free energy models. And today, artificial intelligence and machine learning is being used in various steps in molecular docking as an alternative to the traditional models or traditional statistical methods. So we have several applications. As I said before, we have the drug discovery and development, analyzing the specific binding mechanisms, as we said, virtual screening for several candidates, toxicity prediction, agricultural chemistry, environmental chemistry, environmental biology, and so on. Or maybe we can use it to, for example, induce mutagenesis on the molecules to see how mutations affect the interactions. Sometimes it can be used to design de novo targeting molecules, de novo design of targeting molecules, and so on. So looking at the docking approaches, we have the shape complementarity approach. This approach is basically based on the geometry, like the geometry underlying the interaction. And it uses like several statistical methods to map the geometry of the ligand to, for example, to the receptor, and it looks at like the best geometrical overlap in between them. So it describes the proteins and ligands as complementary surfaces using, as I said, geometric matching. Molecular surface descriptors, we look at the surface. We look also at the hydrophobic region, the hydrophilic region, the solvent accessible regions. So it's usually fast, but like it limits the flexibility of such interactions. Usually real biological interactions are not just like rigid lock and key mechanism that is solely based on a predefined complementarity. So this is one of the issues. Simulation-based approaches, it's basically searching through a whole space of different orientations, for example, and we measure the energies of these different orientations. As you can see here, it stimulates the docking process by calculating pairwise interaction energies as the ligand moves into the protein site. So we can imagine the ligand changing its position, and each time it calculates its energy, and it finds like an optimal position through which this kind of Gibbs energy is minimized, which is the most favorable state. And we'll see later what kind of algorithms can solve this problem. The problem with it is that it is computationally intensive and computationally expensive, and thus it takes like a greater time for the docking to take place. Okay, looking at the types and mechanics of molecular docking, we can classify molecular docking based on, for example, the binding site. Okay, we can classify molecular docking based on the binding site. We can classify molecular docking based on the molecules that are involved, simply on the flexibility of such interaction, as we said, rigid and flexible. And we can have some specialized types of docking, for example, solvation-based docking, for example, metal-ion interaction-based docking, and so on. The mechanics, we rely on scoring functions, we rely on a certain defined force field governing the interactions, and we rely on a search algorithm or an optimization algorithm to find this pose with the lowest energy state. So as you can see here, we have algorithms. They could be systematic, they could be random and stochastic sometimes, and we'll talk about this algorithm, to be specific, the genetic algorithm, later. The scoring functions, they could utilize, they could be based on for example, a theoretical force field describing the molecular interactions within a system. It could be empirical, based on certain experimental empirical data, or it could be like a knowledge-based. And then we assess the docking using certain evaluation metrics. So looking at the first classification category, which is the binding site, we have the blind docking and we have the target docking. And in the practical session, hopefully we'll like try blind docking and we'll try target docking. So from the name blind docking, we basically don't know where is the binding site of the molecule. So the search space is the entire receptor or the entire macromolecule technically. The binding site is unknown. It's not reported in the literature, or maybe we want to identify some novel binding sites. But we'll find later why this blind docking mechanism, like sometimes it's not completely blind, technically. Computational cost, it's high. Why? Because we have a larger search space. As we expand the search space, we increase the number of possible poses and interactions, and thus the computational cost and the computation cost increases. This is why we look for more efficient algorithms and like theoretical computer science to deal with such intensive tasks. So it's a broad exploration. The target docking, we know the binding site of the molecule, or for example, the active site of the molecule is known in the literature. So as you can see here, we just specify the area of the binding site. And after docking, we can know the specific binding mode that is favorable for a certain ligand, and we can uncover the interactions. The molecules involved, fortunately, docking allows a wide range of molecules to be docked. As we said before, we have peptides, proteins, metal ions. But the problem is that with each molecule, we have to rely on, for example, different types of force fields. And sometimes, for example, in the case of metal complexes and metal ions, sometimes you have to devise your own force field, which needs a good theoretical background in the field. Today and tomorrow we'll be working on the small molecule ligands, small chemical molecules, simple molecules. So as you can see here, we have molecular docking of DNA with the heterosteroid molecule. On flexibility, we can divide, as we can see here, rigid or induced fit. So it could be rigid kind of docking, like it follows the lock and key mechanism. Fixed ligand, fixed receptor. It could be semi-flexible. Sometimes we could have a flexible ligand, but a rigid receptor. So in the case, for example, of a transmembranous receptor, so we can assume that it's rigid and the ligand is, for example, flexible. Fully flexible docking, both can be flexibles. Sometimes some certain side chains, for example, flexible active side chain only, active sided chain only, and so on. So here's like a representation. Here we can see like the rigid docking. And here we can see a kind of flexible induced fit kind of docking. Okay, the mechanics and search process. As we said, in molecular docking, we have the searching function and the search algorithms, and we have the scoring function. We could have force field, empirical knowledge, or sometimes a consensus scoring, which is based on a combination of all of the different scoring functions. For the searching functions, we'll see different algorithms, and we'll have a focus on one algorithm. Okay, so defining the force field, it is simply like a model, a mathematical model governing and explaining the potential energy of the molecular system. It takes into account the different intermolecular interactions taking place inside the system. It takes place the different types of molecular transitions, the bonding, the angles, the van der Waals interactions, the electrostatic interactions, and so on. So as we can see here, the purpose of the force field is to evaluate the binding pores, and we want to know the potential energy of the system, so we can calculate the change in this kind of energy upon binding and before binding. We have intramolecular interactions. Intermolecular interactions are taken into account. Common force field, amber, is a very common force field. Also, CHARMM is very popular. You'll see those two. Okay. So basically, as we said, Force fields sometimes can be empirical. They can be fitted to certain experimental data that we have. Some force fields can be quantum derived, so they work at the quantum level, and these can be more accurate sometimes. Looking at the scoring function, we have like different types. Like when we say the scoring function is usually, it doesn't have to be associated with the force field. Force field is a method for some scoring functions. Scoring functions, as we said, it can be empirical also, or it can be consensus or knowledge-based. So force field, basically force field mechanics is just one way of having like a scoring function, and it's a common way actually. And AutoDock, which is the software that we will work with, works using a force field kind of mechanism, which takes into account the van der Waals interactions, the electrostatic interactions, as we said, and so on. And it calculates finally the Gibbs free energy change, which is the final score, which is simply the energy of the system complex minus the energy for individual protein and ligand molecules. So it's a difference in energy, basically. So yeah, this is the main idea. Like we get the score, which could be a theoretical estimation of the delta G. Might not be like a very accurate estimation of the real delta G. It is a theoretical estimation, and molecular docking or these force field kind of scoring functions are not the best actually to get the, for example, the change in energy. Recently, there are some machine learning methods that can be incorporated in the process for more accurate estimation, or as we said before, molecular dynamic simulation. They can be more insightful. Force fields are limited. It correlates with the binding energy. It's not an exact representation. We ignore solvation effect. We usually ignore the dynamic environment surrounding such molecules, while in, for example, molecular dynamic simulation, we take into account the solvent surrounding the molecule, and we create this kind of like mini micro environment. For the search algorithms, we have different search algorithms for different types of docking. We have search algorithms that are more suitable, for example, to rigid kind of docking, which is usually based, the rigid kind of docking is usually based, for example, on shape matching and shape complementarity. While in flexible docking, we have other algorithms. Genetic algorithm is very, or the GA is very popular, and we'll use it in AutoDock. It's basically used for flexible, semi-flexible models, but let's have like a quick focus on the rigid docking algorithms. Fast Fourier transform is well known. Uses shape-based or energy-based grids to match ligand and receptor. So it kind of deals with the ligand and the receptor as spaces overlapping with each other, and we want to find the best maybe kind of overlap between them. We have some systematic processes such as incremental. This is a systematic non-random process such as incremental construction, which decomposes the ligand molecule or the molecule that we want to dock into, for example, different like pieces or fragments, and we dock them systematically or incrementally, and then it reconstructs the ligand in the binding site finally, following certain constraints. Will not violate the basic electrochemical and laws underlying such interactions. For the genetic algorithms, it is a random stochastic or heuristic function for those who know a bit in computer science, not necessarily finding the most optimal solution. It's not like a brute force algorithm. When we say brute force algorithm, we look through the whole search space. For example, you can see here, we have here the search space. Following a fully brute force algorithm, we usually search through the whole search space. And this sometimes is not feasible, especially if our ligand molecule and the protein molecule are very flexible, and especially if the search space is very large. this can be computation very expensive. So we resort to some kind of algorithm that follow this kind of heuristic approach where they want to optimize their current state. Maybe the final state that they end with is not the most optimal, but it is a local optimal state. Maybe we can say it's not a global optimal state. So we have different, if we have, for example, evolutionary computing, genetic algorithms are a part of evolutionary computing. If we see like a basic representation of how genetic algorithms work, so we can basically represent the maybe the poses or the orientations or the conformation of the molecules in terms of genes and chromosomes actually. And we know, like in the theory of evolution and natural selection, it is basically a natural optimization process that is taking place. So what takes place in genetic algorithms is that they mutate their like as we can say, like quote unquote, like genomes or chromosomes, and they keep on measuring their fitness. There is a kind of a fitness function. They keep measuring their fitness, and upon reaching a certain level of desired fitness, they stop, for example. So as you can see here, the gene represents a variable defining the particular aspect of the ligand's conformation, taking into account the translation, the rotation of the molecule, and even the angle of the molecule and the whole pose like of the molecule. We can see here the evolutionary steps in such genetic algorithms, like we have initial library, selection, crossover, and point mutation, and repeat. So this is what's actually taking place in the basic like evolutionary natural selection, Darwinian evolutionary theory. So it's like nature-inspired, bio-inspired algorithm. As we said before, also we have the Fourier transform. Fourier transform used for rigid docking. As you can see here, we are dealing with triple integrals, so basically dealing with kind of like volumes. And like it's multi-step process that is based on shape complementarity, electrostatic potential, hydrophobicity, and so on. Okay. As you can see here, like the receptor, the ligand, they are in a certain space. It scores. Is it finished? No. So there is like a kind of an algorithm governing it. Okay, I think we had a good like insight into the process. Flexibility and high degrees of freedom for molecules creates a larger search space. So for example, if the ligand molecule has a lot of rotatable bonds, has a lot of torsional forces, and so on, we technically increase the number of possible binding poses within certain constraints. And we increase the search space, thus slowing the process. So this is an important thing to consider. This is why, for example, protein docking takes longer time than, for example, may take longer time, for example, than a simple molecule protein docking, and so on. Dealing with different higher structure, this is a very important, like it's a theoretical aspect, but at the same time, it's practically very important to consider. Proteins can exist in different, like, for example, quaternary structure states. They could be multimers. For example, looking at hemoglobin, they could be dimers or multimers, which includes dimers. They could be homomultimers. The monomers or the single subunits are identical, for example. And the binding site could be within the subunits themselves. Sometimes the binding site is at the interface, between the different subunits, and it's not just within one subunit. Sometimes we could have heteromultimers with binding site at one of the monomers, or heteromultimers with binding site in between or at the interface. So thinking logically, if we have a homomultimer with the binding site at one of the monomer, and we just want to understand how this ligand interacts with the subunit, so we basically just take one of the monomers or one subunit, and we delete all of the other subunits. In the case of homomultimers with binding site at the interface or between, we have to dock the whole structure. because it could be something in between. It's neither completely for subunit 1 or subunit 2. As you can see here, for example, we could have this kind of interface, and the molecule can bind here, for example, not, for example, here or just here. Heteromultimers with binding site at one of the monomers. So we actually dock the chain only with the binding site. So if we have like the subunit A and we have the subunit B, for example, and the binding site, we know that the binding site is at subunit A, so we just take the subunit A and dock it. Heteromultimers with binding site between, we also dock the whole structure. How to know about the binding sites? The binding site could be previously known from the literature. If it's not, we have some certain algorithms, binding site prediction tools. We'll see one. And it will score the different binding sites and will give you the most probable binding site and probably the most druggable binding site that is suitable for druggability and targeting by drug. Or we can dock, if we want to know also if the binding site is at the interface or at a single molecule, sometimes we can, or at a single subunit, we can dock the whole structure, and we can dock individual subunits, and we can compare the scores, or for example, the change or the binding energies. So if the most favorable interaction was with the whole structure at the interface, so it's probably at the interface. If for example, it was most favorable at subunit A, not subunit B, for example, so probably the binding site is at subunit A, so we dock subunit A. Protein and molecule databases, PDB, which is very popular with the protein data bank. It has some certain like crystal structures, and you can derive even AI predicted structures, AlphaFold structures, and so on. Proteins, nucleic acids, and complexes. We have the UniProt, also it's for protein sequences. And we have like chemical databases for small molecules. For example, the PubChem, which is popular for small molecules. So you can see here, for example, the PDB. We have usually the PDB ID of a certain protein. And you can see like further, you can download the file, download the PDB files. You can assess the crystal or the X-ray crystallography structure and assess its quality and so on. The importance of crystal structures. Why do we need the crystal structures? It could be simply we need the crystal structures for the docking process. So instead of relying, for example, on AlphaFold predicted, if we have the crystal X-ray crystal structure, we could rely on the crystal structure for the docking simulation for the macromolecule. Accurate binding site definition, and as we said, usually the binding site can be defined in the literature. Validation of the docking results. This is a very important step. In docking, we have docking, and we usually have another term which is known as redocking. Redocking, it's basically like testing our like parameters, testing our software, testing our docking settings, whether they are reliable or not. So we have, for example, a crystal structure with a known ligand that is bound to it. We dock this, like we remove this ligand from the crystal structure, and we redock this ligand using our docking parameters or docking settings, and then we compare the two, considering the crystal structure as the gold standard, using certain evaluation metrics, using certain difference-based metrics, such as RMSD, for example, or the mean square deviation. We can know if our parameters or our docking settings or our softwares are reliable for future docking. So it's like a validation or a testing process known as redocking. We could incorporate cofactors in both molecules and so on. So as you can see here, we have NMR spectroscopy, which is a popular technique for retrieval of crystal structures, or we could have X-ray diffraction, X-ray crystallography data, and sometimes like new research is showing techniques combining both of them for enhanced accuracy. In molecular docking, it's also important to assess like maybe the protein structure, the X-ray structure, to assess the quality of X-ray structure, so we can assess the resolution. We want high quality resolution, so it's less than, for example, 2 angstroms, acceptable around 2 to 3 angstroms. The lower the angstrom, so it's greater kind of resolution. The R free value, for example, less than 0.25, or sometimes you could have a kind of like a ratio measurement. You can see in the literature like different maybe rules or recommended like cutoffs for these metrics, and maybe you have to cite them in your publication. And we have the Ramachandran plots. The Ramachandran plots, they basically show us what kind of orientations and what kind of angles are favorable, and what kind of angles are allowable for this protein structure. So if the protein or some of the residues in the protein show in energetically unfavorable regions or unusual regions, so this raises a concern about the quality of such structure. Maybe you have heard of like the phi angle, the C angles, like we have here with the alpha carbon of the amino acid, the alpha carbon like with the carboxyl carbon, we have the C angle for this bond. We have the alpha carbon with the nitrogen, and we have the phi angle. And these have a degree of freedom. These two angles have a degree of freedom. So here in this plot, we basically compare the phi with the C. So having both of them, for example, 0, is quite impossible, because we will have a lot of like steric hindrance, as we can say in chemistry. So basically, some like sets of phi and C angles are not favorable. But for example, here in the red zone or in the dense zone, these are the most favorable zones. For example, beta sheets can exist here, or sometimes collagen helixes, for example, can exist here. While in this region, it's almost impossible, except if the structure was derived in a wrong way. For docking assessment, we have different kinds of metrics to assess docking. We have accuracy. Correlation is very important with the experimental binding affinity and the experimental data. We have also the RMSD, which is very important, or the root mean square deviation, which is basically like summing the squares of distances between, for example, our predicted molecule and our gold standard. So it basically evaluates the deviation of the predicted ligand posed from the experimentally determined structure. And this is important, for example, in redocking process. We have simply the binding energy scoring and how it correlates with the experimental binding affinities reported, and so on. We have lots of docking tools. In our practical session, we look at the AutoDock 4. We look at the AutoDock Vena. They have some like differences. AutoDock Vena is usually faster. AutoDock Vena is usually more preferred for like high throughput screening of several candidate ligand molecules. AutoDock 4 is usually used like for single docking settings, like not screening. We'll try to. We have the AutoDock Gold. It's also well known, Glide, and so on. But these are very popular. Okay, I think this is the most important key of information. So basically, for the certificate, you can like scan this QR code before we proceed. 2 minutes. Okay, I think we can proceed. We have different like aiding tools we can use in molecular docking. For example, PROTOS predicts and optimizes hydrogen atoms. Like you'll see later that in docking, we have to optimize the hydrogen bonds, we have to optimize the ligand molecules, we have to add the hydrogen bonds, and so on. So some tools can do this process. But sometimes you can find such tools built in within the software you're using, such as AutoDock or anything like that. We have predictive tools, and these are pretty important. We have, for example, the dog site scorer, which is based on like a machine learning approach, like the support vector machine algorithm. What is good with this tool is that it predicts the binding pocket. It scores different binding pockets, and it gives you like a score, a druggability score for the binding pocket, and the accuracy is around like 83 to 88%, I think, for the druggability scores against like experimental data. So yeah, it's a pretty good tool. We have some certain analytical tools. POSView, for example, you can upload, for example, the docked ligand-protein interaction or ligand-protein docked complex, and it creates a 2D diagram of highlighting like the key interactions, the hydrogen bonds, the van der Waals interactions, electrostatic interactions, and so on, for publication-ready figures. We have POSEDit, and we have like other tools. We have, for example, PDB sum, PLP, leg plots, and so on. We'll see them hopefully later. And we have some like auxiliary tools. For example, Activity Finder, it links the results of docking to experimental data. This, for example, the PPI tool is very important in case you aren't interested in, for example, protein-protein docking. PPI is protein-protein interactions. Docking is one of the like techniques that is used for protein-protein interactions. So the high PPI classifies and analyzes protein-protein interaction surfaces, and so on. So there exists like a lot of tools. It depends on like your specific application. So just don't limit yourself to the software you're dealing with. The general workflow of docking, we have the protein and ligand selection. We get, for example, the PDB files, the SDF files of the proteins and the ligands. For example, the protein ligand preparation, we usually add the hydrogens. We optimize the ligand molecule, for example, the simple molecule. So it is at the like best geometrical pose or geometrical orientation, selecting the proper tautomers, proteinomers, and stereoisomers, and so on. Check the protonation states, check the charges. All of these factors can affect the results of docking, so they are important to be taken into consideration. The binding site definition, we can predict it. It could be experimentally derived. Sometimes we have like to remove additional non-standard molecules, such as water, and sometimes we have to keep the water if it is an intrinsic part of the molecule. But like, for example, additional water molecules from, for example, the PDB file or from the crystal structures have to be removed. And then we do the searching process, the mechanics of molecular docking, as we said before, the scoring and so on, and then the evaluation. So as you can see here, we have the target structure, PDB download, the binding site, which could be the active site, a cryptic site, or even allosteric site investigation, allosteric site kind of docking. For the ligand, we have to calculate the charges. So we have the ligand at the optimally charged molecule using some certain acid-base calculations, such as pKa calculation. We have the docking program for sure, which is AutoDock in our case. Verify and standardize before you start using redocking techniques. scoring, ranking, interaction analysis using some auxiliary tools or using some, for example, additional tools, as we said before. And we can transition finally to the molecular dynamic simulations for further analysis. So basically, we have different files that we'll deal with. It depends. You might need these files. It depends on what software you're using. Some softwares are more user-friendly. For example, working with, for example, AutoDock Vina on a certain, for example, browser such as the UCSF Chimera browser. It like it makes a shortcut through a lot of steps, so you don't have like to see even sometimes the GPF file or the DBF file. But for example, in the case of AutoDock, no, you have like to generate each file separately. So we have basically the input files that we have, the PDB files, and sometimes for certain software such as AutoDock, we have to convert them into, for example, PDB QT files. What differentiates PDB QT from PDB is that it contains the charge information. And we have the ligand. We have the grid parameter file, which defines the grid or the search space for the docking area. We have the docking parameter file, which specifies the algorithms and any docking settings. And as we said before, these docking parameters can be optimized using redocking. And the output files, we have the docking log file or the DLG file with the different orientations or the different binding forces and the energies. And sometimes in the case of certain softwares, it can give you the estimated inhibition constants. And you can also get the PDB QT of the whole complex. And as we said, this like PDB QT, and then it can be converted to a PDB file. You'll have a PDB file of the final docked complex with the most favorable state. And you can analyze this like final docked complex using, for example, pose view to create such 2D diagrams. And you can visualize this docking complex for further studies. We could have also high throughput screening. We'll not discuss high throughput screening, maybe in a later workshop. So you basically have, and usually in high throughput screening, in order to automate the process, like instead of working with each ligand, like if we have, for example, a candidate 100 ligands, we usually automate the process using some programming languages along with AutoDock, such as, for example, Perl, or sometimes you could use, for example, Python, certain Python scripts to automate this docking process. So this is basically the AutoDock 4 workflow, as we said, like the ligands, proteins. We have the PDB QT files. We have the docking parameter files. And finally, we would have the docked ligand file and maybe the final PDB file. AutoDock 4 outputs the estimated inhibition constants also. This feature is not like obviously present in AutoDock Vina. So this is the estimated Ki, and this is, for example, the estimated free energy of binding in kilocalories per moles. The more negative, the more favorable the interaction is. And as we said, further analysis, for example, we can refine the pose on a more local search space. So for example, if we dock, if we do some blind docking, we will have like an output of like a probable area where for example, the ligand could bind, and then we can refine our docking on like a smaller search space. So it gives us a more accurate representation. We could visualize the interaction, as we said, using, for example, Discovery Studio, lig plots, PLPs, Protein Plus. Protein Plus is actually like a web server with lots of different tools, including the pose view, including the scorer, the binding site prediction tools, and so on. So it's very, very useful. Molecular dynamics, as we said, this is, for example, a molecular dynamic simulation, and we usually have like an environment that's taken into account. For example, here we have a lipid bilayer, for example. And usually molecular dynamic simulation, it provides more accurate theoretical binding energies. Experimental validation, as we said, the mutagenesis assays, and so on.